My name is Liz Costello. I'm a research scientist at Brown University, and I'm going to be presenting on some recent work I've done with Dr. Joe Braun at Brown, looking at how exposure to endocrine disrupting chemicals might occur through consumption of ultra-processed foods. All right, and so just to start, I have no conflicts of interest and nothing to disclose around that. So just as an overview of what I'm going to be talking about today, in case anyone's not familiar, I'm going to start with some background on endocrine disrupting chemicals and what they are, and then go into a definition of what ultra processed foods are and what I mean when I say ultra processed foods in the context of this research. Then I'm going to describe a new method that we have to predict the overall level of food processing in the diet called FPRO, and then how we've applied that to two different cohorts in pregnant women and adolescents. And then I'll close with just some future directions that we hope to take this. So to start with endocrine disrupting chemicals, or EDCs, these are any chemical that can interfere with the endocrine system, and this can happen in a few different ways. So anything that disrupts the synthesis, secretion, transport, metabolism, or other actions of hormones. This can result in several adverse health effects. Past and recent research has shown that this can increase the risk for reproductive health problems, hormone-sensitive cancers, neurodevelopmental and behavioral problems in children, immunological effects, and adverse birth outcomes. When we talk about EDCs, we're usually thinking about two different broad categories, persistent and non-persistent EDCs. So persistent chemicals tend to bioaccumulate in humans, animals, and the environment, so they can be very difficult to get rid of from your body and difficult to destroy naturally. The half-lives can be several years long, so when you're exposed, these chemicals really remain in you for quite a while after that initial exposure. With non-persistent EDCs, they don't really bioaccumulate in the same way. They tend to be metabolized much more quickly, usually within hours to days. But typically with these chemicals, we're continuously exposed. So they may be things that we encounter in everyday life, almost every day or weekly. So exposure is pretty continuous. In this talk, I'm going to be focusing on just a few groups of persistent and non-persistent chemicals. For persistent EDCs, I'm going to be talking about PFAS, per and polyfluoroalkyl substances, sometimes known in the media as forever chemicals. These are persistent pollutants, so we usually measure them in blood since they'll stick around a bit longer. And PFAS are used primarily in water and grease-resistant products. So this can include things like waterproof clothing, stain-resistant fabrics, non-stick coatings for pots and pans, cosmetics, but also in things like firefighting foams and industrial products. So there are several thousand chemicals in this class, but we really only talk about a few named ones just because there are so many. They're detected worldwide in the environment and in almost all people. Right. For non-persistent EDCs, there's a couple different groups here. I'm going to be talking about bisphenols, which includes BPA, which many people I think have heard of. These are used in plastic manufacturing, BPA and its alternatives, BPS and BPF. They can be found in the linings of cans, in bottle caps, in paper receipts, in pipes, and other products. Parabens are another non-persistent group of chemicals typically used as preservatives. And while they're not frequently found in foods, they're most often found in cosmetics, pharmaceuticals, and other personal care products like shampoo, lotions, and sunscreens. And then finally, I'll be discussing some associations with phthalates, which are used as plasticizers that can increase the flexibility, transparency, and durability of plastics. They're most often used in PVC plastics, but that means they can also be found in medical devices, toys, vinyl flooring, and other household products. So the original phthalates are orthophthalates, which includes chemicals like DEHP, but there are also alternatives, chemicals with similar structures that are not technically phthalates that may include DEHTP and DINCH. So exposure to endocrine disrupting chemicals has been shown many times to have adverse effects on maternal and child health. So prenatal exposure to EDCs has been shown to be linked to altered hormones, to higher cholesterol, to gestational conditions, and to adverse birth outcomes. In kids, both prenatal and childhood exposure has been linked to increased risk for neurodevelopmental disorders, metabolic conditions, and reproductive health issues. The mechanisms underlying this might be a few different things. They might include epigenetic reprogramming, disrupted hormone signaling, increased inflammation and oxidative stress, or placental dysfunction as a result of EDC exposure. Okay. So to pivot now to ultra-processed foods, this is a little bit of a fuzzy concept and is also a hot topic a little bit in the media right now. And there are many different definitions floating around, but the one that most of us agree on at a baseline is that these are products that really require industrial equipment to create. So these are not foods that you can make in your home or in a restaurant. They tend to share the same characteristics. So they tend to be high in calories, high in sugar, saturated fat, and salt, and low in good or beneficial nutrients like fiber, natural vitamins, and minerals. But when we think about how ultra-processed foods might affect health, there are some kind of different considerations that you might want to think about depending on the mechanism that you believe is sort of underlying this relationship. So are these foods healthy? And then why and to what extent are they processed? And what is that processing doing to both the nutritional composition and to any chemical exposures that might occur through the food? And then are there any additives that we think might be causing health effects? But very broadly, almost all the foods in this category share the additional characteristics that they are less satiating, which means that you need to eat more of them in order to feel full. They've been shown to negatively impact the gut microbiome and overall increase the risk for metabolic disease and other conditions. And the term ultra processed really comes from this 2019 commentary from a research group in Brazil, which proposed 4 categories of food processing called the NOVA classification system. So of these 4 categories, the ultra processed category is the most highly processed category, NOVA category 4. And so the goal here was really to distinguish as much as possible between processed foods and ultra processed foods. And then using this method, in the last few years, researchers have been able to link ultra processed food consumption, as I've said, to many adverse health outcomes. But just to orient you to what these are, I'm going to talk a little bit about what each of the NOVA categories are. So you can see for yourself sort of how these might get a little bit fuzzy and why we might want to explore different ways to classify ultra processed foods. So group 1, the least processed category, are foods where there's really nothing added to the original food. And any processing methods tend to be pretty simple, things that you can do in your kitchen. So pre-chopped produce that you might buy at a grocery store, dried fruit, frozen foods, things like that. The examples tend to include things that you would buy from the fresh or dried bulk section of a grocery store or a farmer's market. So fruits, vegetables, legumes, dairy products, herbs and spices, yogurt, plain yogurt. And then when we get to group 2, we get a little bit more processed. So these are culinary ingredients and not really whole foods. And so there's been processing happening here by nature. But the purpose of the processing is really to cook group 1 foods. And so these include cooking fats, natural sweeteners, cornstarch, salt, and other minerals. In group 3, these are the result of combining group 1 and group 2 foods. And so ideally, this is the group that you would see most of your meals fall into. Any processing that's happened tends to be canning, bottling, fermentation. So we're looking at canned beans, bottled juices, smoked and salted products, and then fresh bread, pastries, things like that. But the final category is the ultra processed food category. And I've included a quote here from the Montero paper that really highlights the emphasis on industrial processes and industrial uses of different ingredients and formulations in order to create these foods. And any purpose of the processing here is really to create a profitable product. So it's not really preservation, it's not really taste, it's can we make something that will sell to the largest number of people in a way, especially that will displace other food groups. So the ultra processed food category is really a category designed to have you eat more of this and less of groups 1 and 3 foods. The processing methods used here look a little bit more like a chemistry lab and a little bit less like a kitchen. So these are really, again, foods that require sophisticated equipment and technology that you won't be able to replicate at home. We usually have additives and non-culinary ingredients involved here. So things like high fructose corn syrup, emulsifiers, preservatives, and things like that. There's sort of a rule of thumb that if a food creates or contains 5 or more added industrial ingredients, that's the line where we would say it's ultra processed. And then the packaging, again, because these tend to be foods produced by companies, very sophisticated. We can imagine what most of these are. It includes things like soft drinks, packaged snacks, anything labeled instant or nuggets, usually cake mixes, breakfast cereals, meal replacement shakes, protein powders, and that sort of thing. But there has been a lot of criticism of the NOVA system. So it certainly has limitations, the largest of which is that it's very difficult even for experts to agree on which foods fall into which NOVA categories. So multiple nutrition experts, when they're given the same food and the same ingredient list, will make different decisions about whether or not a food belongs in NOVA category 4 or 3. There's also this question about whether all the additives involved in these foods are really created equal. Many are considered to be safe. Some have received a lot more attention recently as being associated with potential adverse health effects. But a lot of these additives occur in combination, and so it can also be really difficult to determine if this is, the source of a health problem. Many plant-based foods that we see nowadays are also considered ultra-processed, even though they're recommended for people trying to pursue a plant-based diet or recommended for people who are unable to drink milk or trying to avoid animal products in general. And so these can often have favorable nutritional profiles, even though they still fall into this ultra-processed category. So not all of our concerns about the health effects of ultra-processed foods are really reflecting the same core issue. So are they healthy for us? Do they have these additive ingredients that might be associated with health issues? And then what we're interested in and what I'm going to talk about in this presentation is whether or not there might be inadvertent contamination of these foods with chemicals like PFAS, bisphenols, phthalates, and parabens. But because of all these issues with the NOVA system, there have been some new methods to classify the degree of food processing and try to classify foods as ultra processed versus not. So our collaborator, Julia Menichetti from Harvard, has tackled a little bit of the reproducibility issue with this new machine learning algorithm that can predict the degree of food processing as a continuous variable. So instead of having different researchers take a list of foods and categorize them into NOVA category 4, 3, 2, or 1, you can use this machine learning algorithm to do that for you. It was trained on NOVA categorization still of USDA food databases. So basically these databases have a food, has the nutritional composition of that food, and then that food has been assigned a NOVA category. So the prediction in the algorithm is based on the nutrient composition of each of these foods, under the assumption that ultra-processed foods have a unique nutrient profile that reflects the degree of processing in a way that you wouldn't find in a whole or a natural food. And so Dr. Menichetti applied this to adult dietary recalls from the National Health and Nutrition Examination Survey, just to look at the overall degree of processing in the diet and also to link that with chemical exposures and health outcomes. And I recommend reading her paper for more about that. But one advantage of the NOVA system is really that it emphasizes the ability to replace more processed foods with less processed foods. So this is an example of a figure I borrowed from Dr. Menichetti's paper. On the y-axis of this figure, we have an FPRO score, the degree of food processing of each individual food. It ranges from 0 to 1. And in this example, these are all onion products. So the FPRO score ranges from close to 0 for raw onions, completely unprocessed onions, and then closer to 1 for onion rings. But you can also see in this example that onion rings made from fresh products or fresh ingredients have a lower FPRO score, lower degree of processing than onion rings that were prepared from frozen. So you can see how you might be able to use this as someone who wanted to reduce their overall reliance on ultra-processed foods to make swaps. In our studies, though, we're interested in the overall degree of food processing in an entire diet. And for that, we're looking at IFPRO. So sort of a summary measure of all the foods that someone has eaten weighted by their consumption, the amount of each of those foods that they've consumed. So the formula we're using is here, and the definitions for each of these components of the formula is here. But basically, this is just a weighted average, again, of all the foods that someone reported eating. You can weight it by calories or by grams. They have slightly different benefits. So if we weight by grams, you account for foods that have no caloric value. So diet, sodas, things that are artificially sweetened that may be lower in calories, but you still may be eating a lot of them. And then this histogram, again, I borrowed from Dr. Menichetti. This is showing the distribution of IFPRO scores in adults from NHANES 1999 through 2006. So this indicates that a lot of adults in the US, at least during this NHANES cycle, had a diet that was very heavily reliant on processed foods. So the mean here is around 0.8 on a scale of 0 to 1. But this figure also calls out two participants or two individuals, A and B, who have very similar caloric intake, but also very different IFPRO scores. And so we can use the IFPRO measurement to get a little bit more information about food processing independent of calorie intake. All right. So there's potential for EDCs, or for us to be exposed to EDCs through food, and especially through ultra-processed food, because of the amount of packaging and production equipment that food comes into contact with throughout the processing. So food packaging may contain BPA, PFAS, plasticizers, and these chemicals can leach out from the packaging into food, especially depending on the qualities of the food or preparation methods used to create it. So foods with different fat contents or that are prepared at different temperatures may be more or less likely to have these chemicals present in them. And then production equipment used in more processed foods can also be a source of EDC exposure. So things like hoses, storage equipment, gloves, sanitation products may contain plasticizers or antimicrobials. And then because persistent pollutants like PFAS can bioaccumulate in meat, fish, and other animal products, there is this potential for foods that are ultra processed or that have really broken down whole foods and recombined them in unique ways to ultimately concentrate these persistent chemicals in the final product. So given that, we were interested in using this FPRO score, the overall degree of food processing and participants' diet, to look at how this could affect their exposure to EDCs. So we have two cohorts, the Illinois Kids Development Study, or IKIDS, which is a cohort of pregnant women in Champaign-Urbana, Illinois, and then the Health Outcomes and Measures of the Environment, or HOME study, which is a birth cohort, but we're looking at an adolescent visit from Cincinnati, Ohio. And both of these cohorts were enrolled at about the same time and have similar levels of exposure to the US population for these EDCs. We measured the same chemicals in both cohorts, 4 PFAS, 2 bisphenols, a few parabens, and a few metabolites of phthalates and phthalate alternative chemicals. We calculated gram-weighted IFPRO scores, and we did this a little bit differently in both studies. So in the iKid study, we had a food frequency questionnaire, and in HOME, we had 24-hour recalls. And so I'll go into detail about those in the next slides. To start with the iKids cohort, these again were pregnant women between the ages of 18 to 40. Most of them are non-Hispanic white. Most of them were highly educated. And at their first visit, they were less than 15 weeks pregnant. We measured PFAS in serum samples that were collected at a median of 17 weeks gestation, and then biomarkers of bisphenols, parabens, and phthalates in urine using 3 to 5 pooled urine samples. So when participants came in throughout their pregnancy, they provided at least 3 and up to 5 urine samples taken at different time points. And these were standardized to specific gravity for the analysis. The diet assessment was conducted using a block food frequency questionnaire that was completed at the first study visit. And so it assesses the past 3 months of dietary intake, so roughly the first trimester of pregnancy. For anyone who's familiar with an FFQ, it's structured in a way that you respond to a set number of questions. So you respond to questions about, did you eat a food and then how often did you eat it, and how much did you eat? And then sometimes there's additional questions about preparation of the food or about things like whether it was low fat, if it was fried, um, occasionally some other details, but overall it's very broad with very limited details. So our assignment of FPRO scores got a little bit more involved. But basically what the FFQ was doing is it's built on some assumptions about which foods are most commonly eaten in a reference population. So in the block FFQ, each question is a weighted average, basically, of the most common foods eaten in the US. And so this is used to calculate nutrient contributions to the diet, and we use the same sort of method to look at FPRO. So as an example, we have an FFQ food, apples. The apple food is really a composite of several individual foods. So for apples, we might have raw apples, fried apples, baked apples. And each of these individual foods is assigned an FPRO score. So these can be very low for raw apples or much higher for fried apples in this example. And then we took a weighted average of all of the component foods of the apple FFQ item. So these are weighted by the overall grams consumed per day in the US population. And what that gave us in this example is an FPRO score of a very low FPRO score of 0.04. And that's really because when you look at this, and the grams consumed per day, most people in the US, when they eat an apple, are eating a raw apple, which has a very low FPRO score. And so that's what's reflected in this composite. So we did the same thing for all foods in the food frequency questionnaire. There were about 184. And then this figure here is just showing you the distribution of each individual food that we had from the FFQ and their FPRO score. So these box plots, in these box plots, the mean is the solid line, which you can see better in some of these than in others. And then each gray dot is the individual food FPRO score. And what we see is that the most processed categories of foods tend to be mixed dishes, grains, and snacks and sweets, and the least processed category is fruits. And then there's a little bit more of a range going on for beverages, vegetables, milk and dairy, and protein foods. From there, we calculated the IFPRO score for our iKids participants, which is, again, the overall degree of processing in a participant's diet. So we had a mean here of 0.54, which was pretty good. And the way we calculated these for participants, I'll give you an example, is in this table, we have the first 20 or so FFQ items and their corresponding FPRO score. So these are color-coded based on how processed it is. More red, higher processing, green, lower processing. And we looked at the grams of each food that participants reported eating on average. And so what you can see even in this example is that participants with the highest FPRO scores, so participant E here, is really not eating a lot of fruits and vegetables. Very limited intake of fruits and vegetables. And you can see sort of higher intake of things like French fries, pancakes, egg sandwiches relative to the other participants. And then those with the lowest FPRO scores in this example are eating the most fruits and vegetables. So we summarize this across all participants to get the overall degree of processing. And then this visual is really a sanity check. We're sort of seeing what we expected to see. Everyone who is eating more apples tends to have a lower FPRO score. All right. For the analysis, we use covariate adjusted linear regression. We adjusted for demographics and potential confounders, in this case, parity, stress, and energy intake as calories. And then just to note that when we did this analysis, we log transformed our chemical concentrations and scaled our FPRO scores. And so what that means is that when I show you the coefficient plot on the next slide, these estimates are really expressed as the percent difference in EDC concentration per standard deviation increase in FPRO. So a standard deviation in this case is about 0.14. So these are the coefficient plots. We're looking at the associations between FPRO and PFAS right now. The dot here is the effect estimate for the IFPRO EDC association, PFAS association, and then the line is the confidence interval. So we see a positive association between overall degree of processing and PFOA, which is a legacy PFAS that was used for many decades though it was voluntarily phased out in the early 2000s, but it does stick around as a persistent pollutant. So for each standard deviation increase in IFPRO and iKids, we saw that PFOA concentrations were about 7% higher. When we looked, though, at bisphenols and parabens, we really didn't see any statistically significant associations, though we do see a few associations in the directions that we expect. So we find positive associations with BPA, and we expect that because we know that BPA is still used in food packaging, so it makes sense. With ethyl paraben, this paraben is not typically used in any food contact materials, not typically found in the diet, and so it's good that we don't see a positive association, since it's more of a negative control. With the phthalates and phthalate alternatives, we see a lot more positive associations. So we see links between IFPRO and a few of the older phthalates, DIDP, DNOP, and BVP, or their metabolites. And then also with the phthalate alternative chemical DEHTP. And this is all consistent with previous studies in adults that have looked at the NOVA4 category foods as an exposure and linked that to endocrine disrupting chemicals. Specifically, these phthalates appear in previous literature and in our study, indicating that the FPRO score overall is doing a pretty good job of approximating NOVA4 as we understand it. All right. Pivoting to the home study, our home study participants were enrolled at about the same time as the iKids participants, but they are adolescents. But they were also mostly non-Hispanic white, and most of them had mothers with at least some college education. We measured PFAS again in serum, but the urinary metabolites and biomarkers we measured this time in just a single urine sample. So only one urine sample, and they were creatinine standardized. The diet assessment was done using 24-hour recalls in this case. So for a 24-hour recall, we asked participants about everything they ate and drank the day before. So we asked them, what food did you eat at breakfast? How much of that food did you eat? And then occasionally there's more detail available, such as the brand name of the food, if it was branded, any preparation methods, and then the source of that food is occasionally also available. The recalls in home were done on 2 weekdays and 1 weekend day. And so you take the average of the 3 recalls to estimate usual dietary intake. So we use that estimate for the PFAS associations because PFAS are persistent pollutants, and we're really looking at overall diet and not something more recent. For the urinary biomarkers, because these non-persistent EDCs have much shorter half-lives and are excreted much more easily, we looked at just the first 24-hour recall, so the most recent dietary exposures. All right. Again, we calculated the individual food FPRO scores for each of the foods reported in the 24-hour recalls. There were a lot more in the 24-hour recalls than there were in the FFQ. So we had almost 2,000 individual foods. You can see a lot more gray dots on these box plots. But we see similar patterns to what we observed with the FFQ foods, which is that the most processed categories tend to be the mixed dishes, grains, snacks, and sweets, where fruit tends to be the least processed category. And then we get a little bit more of a spread for milk and dairy, protein foods, vegetables, and beverages. We saw that there were higher IFPRO scores, so higher degrees of food processing in the diets of the home participants than in the iKids women. So the average here was around 0.7. And these histograms are showing you the distributions for the average of all 3 dietary recalls, and then just for the first dietary recall only. When we look at what foods kids were eating, we can see that these higher scores are reflected in the individual foods that they all reported. So for participants A, B, and C here, participant A really did not report eating anything unprocessed. So this is someone who ate a lot of soda, ate a lot of snacks, whereas participants B and C have a little bit more of a little bit more variation in their overall diets. And so this is reflected in their IFPRO scores too. Where participant A here, who really didn't eat any vegetables or fruits, has a very high FPRO score. And participants B and C tend to have lower scores since they're eating a bit more unprocessed foods. All right. We did the same type of linear regression, adjusting for covariates. So the covariate set's a little bit different here. In the PFAS models, we adjusted for breastfeeding, since in children, breastfeeding can be a greater source of PFAS exposure, and we have that information. We also adjusted for the month of the study visit, so the month that the first recall took place. And this is because depending on whether a child is in school or what season it is, they might be using different personal care products. They might have different exposure patterns to the non-persistent EDCs. Again, we logged transformed our chemical exposure or chemical outcomes and scaled the IFPRO score. But we really don't see any statistically significant associations in the kids. So here are my coefficient plots again. Here we don't really see any consistency with PFAS or with the bisphenols or parabens. There is a indication that there's a positive association maybe with PFNA, which is another legacy PFAS, but we didn't see that in the iKids cohort. For the phthalates and phthalate alternatives, we do see some of what we expected to see. So these are similar magnitude and direction of associations in home as we saw in iKids. It just doesn't really reach the level of statistical significance, which may be due to sample size. We have fewer people in the home study than we do in iKids. But it could also be something to do with adolescents in general. If adolescents have different exposure pathways, or if maybe there's not been enough time for exposure to occur. And so because of that, we wanted to see if we could replicate this analysis in NHANES. So by replicating in NHANES, we can look at a representative population that reflects the US population at roughly the same time that the home kids were enrolled. So in NHANES, I don't have this to show you right now, but there were similar IFPRO distributions. So overall degree of processing in the diet was similar in NHANES adolescents as it was to the home kids. And then the endocrine disrupting chemical concentrations were also very similar in NHANES compared to the home kids. And so these coefficient plots are the results. I'm showing you here the home study in the solid lines and the NHANES adolescents in the dashed line. And overall, again, we see pretty null results with the exception of some of the bisphenols, which appear to have positive associations in NHANES. We didn't really see that in home. And then for the phthalates, everything is going in roughly the same direction, or if it's null in both studies. So this sort of tells us that in adolescence, there may not be as strong of a relationship between food processing and chemical exposures, or maybe that there could be some other things going on. Because the literature is pretty consistent in terms of whether or not ultra-processed foods and may increase the risk for chemical exposure in adults. We see this in many different studies. We see the same chemicals come up as we do in iKids. So in the home study, the adolescents, there are some differences in terms of how the study was designed and also in the exposure patterns that we might expect to see for kids. So we've only had a single urine sample in the kids in both NHANES and in home. And this can kind of limit our ability to detect associations with the non-persistent EDCs. So the urine samples are collected after participants have been fasting. And so it's possible that the dietary exposures are just not recent enough for us to detect an association. There are also limitations associated with the diet assessment. So 24-hour recalls especially are subject to recall bias. And then with kids, it's possible that they don't remember what they ate yesterday. It's also possible that they may not want to report all the foods that they ate if their parent is there with them. And if the parents are assisting with the recall, it's possible that the parents may not know what their child ate either, especially if the kid was at school, if they were at their friends' houses, or doing an activity where the parents didn't see exactly what foods were eaten. It's also likely that diets are not really stable in children. And so when we look at something like a persistent pollutant like PFAS, it's possible that there's not been enough time to really see any associations with food. The kids are going through puberty, their diets are changing, and so they may not have had enough exposure through diet for us to really see an association here. And then for why we see different associations in adults versus children, there's also the possibility that preferential food or food preferences are really different for children versus adults, and access to those foods overall is also very different for a child versus an adult. And so there may just be differences in the actual foods consumed. And we're still exploring that. We're still looking at which food categories and what degree of food processing within food categories might be linked to EDC exposure. All right. And then just to acknowledge some overall limitations of both studies, both of our participant populations are mostly white. They come from middle, upper class, highly educated families. And so our results here may not be generalizable to different populations or for people with different eating habits. As I've mentioned before, we are missing details in the FFQs and the 24-hour recalls that would really let us dig into which foods are definitely ultra processed versus which ones maybe are just not specified, where they came from, how much packaging might be involved, or any of those other important details. We also are really not accounting for different characteristics of different ultra processed foods, including nutrition, whether or not something has more additives than something else, whether or not something is more packaged, needs to be reheated, or any other characteristics. And then finally, the machine learning algorithm that we've used to generate the FPRO score is only really as good as its training data, which is still based on NOVA classification and all of the limitations that are inherent to that. But it is at least more reproducible just because there's only been one instance of classification rather than multiple researchers across multiple different studies making different decisions. All right. So we saw in our pregnant women cohort that consumption of more processed foods may increase the overall risk of EDC exposure. And this is consistent with previous work in adults, both pregnant and non-pregnant, that looked at NOVA category 4 and its relationship with EDC exposure. And so this suggests that the FPRO method works, or that at the very least it's capturing the same information as a manual classification for NOVA. An advantage of the FPRO, though, that we hope to explore more is that it encourages substitutions. So on a personal level, comparing FPRO scores of individual foods may help somebody make a swap to bring down the overall processing level of their diet. And so to borrow again from Dr. Menichetti, she looked at this in her paper at the impact of substituting one dish or 10 dishes with a less processed alternative. And so what these histograms are showing you is that as you make more substitutions, the overall degree of processing in your diet goes down. And in her paper, she also showed that making these substitutions could lower the risk of metabolic syndrome and decrease urinary BPA concentrations. So there's a lot of potential there. And then finally, we hope to keep digging to really identify which specific foods and food groups are increase the risk the most for different EDCs. And then from there, whether we can determine the source of these chemical exposures and really take action to reduce them. All right. So this is the end of my presentation. Thank you all very much for listening. I just want to acknowledge our funding sources. The study is funded by an R21 from NIEHS, and Dr. Braun and Dr. Strakowski are the PIs of this study. I also want to thank Julia Menichetti for giving us all the information we need to calculate FPRO scores, and for Torin Block, who gave us all the information on the block FFQ that we needed to calculate FPRO scores there. And then everyone else who has contributed to this project, really appreciate your input. And my contact information is here if anyone would like to contact me.